Yeah, I think it's also when we discuss like she's been incredibly confused. Like she
Mhm.
does not think this home that she's lived in is her home.
Yeah.
And she wakes up and I'm like, I mean, what if she wakes up and tries to leave the house in the middle of the night? That
Yeah.
almost happened last night. I'm like, oh, gosh, you don't want, I mean, you don't want to push them into, going to home, but it's almost at that point.
Yeah.
Oh, all right, let's finish up. And what I would like to do, hopefully, is finish up 24 today, which probably won't happen. All right, but the thing is, 24 and 25 are so related, especially the end of 24 and the start of 25, that it's okay if we don't finish it completely, but we should get through most of it. Now let's go through in terms of what we see in pancreatic secretions. Keep in mind with the pancreatic secretions that one of the things about that chyme is that the chyme is very acidic. So one of the things we have to do is neutralize that acidic chyme. And we do that by the release of bicarbonate ions from the pancreas. So bicarbonate is going to be released as part of our pancreatic secretion. Bicarb is a base and is going to neutralize the pH of that chyme. Now, by neutralizing the pH of the chyme, pepsin turns back into pepsinogen. So pepsin becomes inactivated. Pepsin is only active then in the stomach, right? Once it hits a duodenum, the bicarbonate gets released from the pancreatic juices through the major pancreatic duct, through the ampullator into the duodenum, and that bicarb will then neutralize your hydrochloric acid. At the same time, it also allows pepsin to reform and reattach the amino acids and deactivate. Now, the good news is that we have other enzymes here that takes the place and finally cuts those really long strands of amino acids. So what I want you to think about is this, right? When you first eat that food, you have like 1,000 amino acids in length, protein, and the protein is all folded really tightly. Once the food goes into your stomach, the hydrochloric acid unrolls it, so then it's no longer folded, and the pepsin starts cleaving it. But once it starts cleaving it, it's still pretty long chains of amino acid. So instead of 1,000 amino acid long, super long chain, now it's maybe, five, 200 amino acid long chains. So it does cleave it, but not fine enough. That's where the other proteases, right, come in. So let's talk about this. Trypsinogen, chymotrypsinogen, procarboxypeptidase. These sound super long. When you see something end with a GEN or, right, sound super long with a prefix that shouldn't be there, procarboxypeptidase. These three are also zymogens, right? And again, a zymogen ends with a GEN, right? Pepsinogen, angiotensinogen. Those are all zymogens, right? Now, they're all produced slightly longer, just like what we saw with pepsinogen. So you have usually between 8 to 10 amino acids that are too long. So you need to cut those amino acids to become active. In terms of pancreatic juices, 1 to 1.5 liters a day is produced, and the pH can be anywhere from 7.1 to 8.2. So just mildly basic to maybe moderately basic. So in terms of the pancreatic secretions, contains water, lots of enzymes, and sodium bicarb. Keep in mind the main enzymes that they produce, especially the main proteases. Proteases are enzymes that break up proteins. So trypsinogen, chymotrypsinogen, procarboxypeptidase, and even proelastase. These are all zymogens produced by the pancreas. Now, in order to activate them, like pepsinogen was activated by hydrochloric acid, well, in order to activate all of these enzymes, we need this one to be active, trypsinogen. Trypsinogen activated by enterokinase. Now, the enterokinase is a brush border enzyme. So this is an enzyme only found on the microvilli of our absorptive cells, our simple columnar cells that are lining the villus. Those simple columnar cells have microvilli, and on the microvilli are receptor enzymes. This is one of the receptor enzymes, enterokinase. And what enterokinase does as a receptor enzyme is that this enterokinase, that is a receptor enzyme, right, attaches to trypsinogen and cleaves trypsinogen to trypsin. Once trypsin's produced, trypsin activates everything else. Trypsin activates more trypsinogen to form more trypsin. Trypsin activates chymotrypsinogen to chymotrypsin. Trypsin activates procarboxypeptidase to carboxypeptidase. Now, these three enzymes cleave the long chain amino acids at different points. So that's how they're different. So you're probably asking, why do we have three different three different proteases here? Well, they are different in where they act. Some of them are going to cleave at the amino acid, the acid end. Some of them are going to cleave at the amino end of our long chains. So they act in different areas, in other words. Now, proelastase, activated by trypsin as well, digests elastic connective tissue. I know we don't want to think about it, but with eating meat, right? When we eat meat, we're going to have, you're eating skeletal muscle. If you're eating skeletal muscle, it's going to have, remember, all skeletal muscles, like ours, they're multinucleated. And if they're multinucleated, they contain lots of DNA. So you need deoxyribonuclease to break down the DNA, the chromosomes in meat. I know it sounds crazy. And then you need them to absorb the DNA bases that we will then use for our own, right, DNA replication. And ribonuclease breaks down RNA, and then those bases we can use for protein. Right, well, so let's continue here. What else? Again, right, if you know what enterokinase is, you'll be in pretty good shape here. So you have all these pancreatic secretions, all these enzymes, trypsinogen, chymotrypsinogen, procarboxypeptidase. The most important activation, though, is what enterokinase does to trypsin. Right? Keep in mind that enterokinase is found on the brush border, the microvilli of our simple columnar absorptive cells. Right? Once trypsin is produced from trypsinogen, trypsin activates everything else. Right? So making it easy, it just activates everything else. It activates and forms more of itself. Right? The trypsin also activates all of the other enzymes, all these other zymogens, right? Now, all of these act at different areas, the proteins, and they're proteolytic. Pancreatic amylase continues the digestion of starch, and this amylase is going to, right, so what happens is in your mouth, the salivary glands contains salivary amylase, which breaks down that super long strand of glucose molecule. Keep in mind, starch is nothing more than thousands and thousands and thousands of individual glucose molecules bound to each other. So thousands of glucose molecules. Salivary amylase breaks it down slightly. Then in the stomach, it stops. There's no enzymatic work on carbohydrates in the stomach. In the pancreas, in the small intestine, the pancreas then releases pancreatic amylase, which then continues digestion of starch. By the time the amylase has done its job, that super long strand, thousands of glucose molecules bonded together, now becomes a disaccharide, all right? Individual glucose molecule bonded with another glucose, right? So when you have two glucose molecules bonded to each other, that's maltose. So by the time we're done and pancreatic amylase has done its job, we will have maltose, which is two glucose molecules. And keep in mind, we need to even finally separate those coming up. Pancreatic lipase digests lipids into smaller droplets of fats, and then the bile will keep those smaller droplets from recombining, right? And that's what we mean by emulsification, separation. Now with bicarbonate, and we're just showing you how we produce it. So what we're gonna see is that here's a blood vessel, here's our cells that are found in the pancreas, and we take the carbon dioxide in the bloodstream, add it with water in the cells, right? In the process, make carbonic acid, and this carbonic acid deprotonates, the hydrogen goes into your bloodstream, where the bicarb goes into the ducts right here. So it's just a way of just producing it. I usually don't ask questions just based on this kind of pictures. It just kind of be kind of very confusing. I wouldn't even say kind of. Right? We talked about bile secretions already in this picture. I think I just re-added it to show you guys. Again, keep in mind that it's CCK and secretin. They're both released when acidic chyme from the stomach gets pushed into the small intestine, specifically the duodenum. As it gets pushed into the duodenum, CCK gets released, which causes the bile to then, the gallbladder to contract. As a gallbladder contracts, then very concentrated bile goes down the common bile duct into the duodenum. Bile is slightly basic, helping you neutralize some of that acidic chyme, right? Now we're going to see secretin causes new bile to be produced. Secretin is going to be very important, though, for the pancreas. Secretin in the pancreas is what's going to cause the release of a lot of your enzymes produced by the pancreas, a lot of your pancreatic juices. Secretin is going to cause production of more sodium bicarbonate. So then you have bicarb that goes through the major pancreatic duct, along with the slightly basic slightly basic bile coming down, the bicarbonate, and the bile will then neutralize that acidic chyme coming in from the stomach right here. All right, in terms of the pancreatic secretions, we mentioned what secretin does. Acidic chyme in small intestines causes increased sodium bicarb release. We talked about what CCK does, which, right, also increases digestive enzyme release from the pancreas. The last thing is this one, GIP, gastric inhibitory peptide. The name should tell us what it does. Gastric inhibitory peptide. Again, this is the intestinal phase, right? Food, that acidic chyme goes into your intestine. Now we're entering a new region. So we need to slow down all the gastric emptying. Gastric inhibitory peptide will help do that. It decreases gastric motility, right? Again, three very important and, pancreatic secretions. Again, I think there's like just one more, gastrin, when we add the stomach. So it's gastrin, secretin, CCK, GIP. Those are the four main hormones released from the small intestines in the stomach. Pancreatitis, inflammation of the pancreas, can occur with mumps. Right? Now with pancreatitis, acute pancreatitis, very, very heavily associated with alcohol intake. Right? Alcoholism causes acute and chronic pancreatitis, sometimes produced by biliary tract obstruction. So then you might have, like we mentioned previously, a gallstone. The gallstone that stone can travel anywhere. So when it gets stuck at the ampulovator, now you have the worst of all possible kind of scenarios. You not only have the gallstones that impacting your gallbladder, but now because of where that stone is residing, it affects your pancreas. One of the things we do check for if you have pancreatitis is this, lipase levels, right? You'll see elevated lipase levels in pancreatitis. Large intestine, here's that ileocecal junction, and the valve is that connection, that small muscle, that smooth muscle, that's like a sphincter, right?
valve, sphincter are used interchangeably. So just keep that in mind when you're reading. It extends from the ileocecal junction to the anus, which is an opening right here. Consists of the cecum, which is a blind pouch start of the large intestine, right? So it ends up like a closed end area. The closed end area of the large intestine is a cecum. Main purpose is the absorption of water and salts, secretion of mucus, and lots of bacterium, gut bacterium that we see here, right? This is crazy. 1.5 liters of chyme enters the cecum per day. 90% of the chyme is reabsorbed. Most of that is water, right? So 90% of that water is reabsorbed, and about 80 to 150 mLs of feces will then be released per day. Normally, obviously, right? Now, what do we see here as well? And we will see in that last slide, as part of our cecum, you'll see this kind of almost like, looks like a little worm right here coming through. That is your vermiform appendix. Your vermiform appendix is your appendix, right? So when you hear appendix, that is your vermiform appendix right here. What else do we see, right? The colon, and keep in mind, your small intestines throughout the duodenum, jejunum, and ileum, you had inner circular, outer longitudinal layers of smooth muscle, 2 very strong layers of smooth muscle. Now we're going to see in the large intestine, it's slightly different. Instead of 2 very, strong, large areas of smooth muscle, right? Because it's got smooth muscle on the outside, it looks like it's like a sausage, right? When we look at the large intestine is sometimes called the colon. Those words are used interchangeably. Well, in the large intestine or the colon, instead of having this really complete outer longitudinal layer, instead you have a little strip called the tinea coli. The inner circular layer is still present right here, but the inner circular layer, right, is arranged into little areas called haustras. So here's a haustra, here's another one, here's another one, here's another one, right? These haustras are pouches, and it's formed by the contraction of both the inner circular and the outer longitudinal layer. Keep in mind again that the outer layer is not complete, right? Because it's not complete, you don't see, it looks like little pouches coming out. Now, some of you might have heard of a diverticulitis or have problems with your diverticula, right? Now, when you have diverticulitis, you have an active infection of a diverticula. What is a diverticula? Is when you have the haustra right here, and there's a little weakness in some of the muscles here. And then instead of having it just extending this little bit of room, right? Because there's weakness, it extends out. As it extends out, feces can get stuck in that region. And then it gets infected and you have diverticulitis. The thing we have to worry about with diverticulitis is that just like in a balloon, what happens if you keep filling up a balloon, right? If you keep filling it up,
I had it before.
balloon material, thins out and it can burst, right, Stephen?
Yep.
Yeah, and that's
I
what
was in the
happens
hospital.
with diverticulitis. You thin out the muscle layer, right, enough, there's a chance, small chance, but a chance that it can rupture. And now you have feces introduced into your abdominal cavity, right? That's not good. So that does happen. I remember seeing it when I was in, going through my general surgery rotation, and it is a mess, right? When it kind of ruptures and leaks out. And now we have to try to find where it is so that we can, and we can't just, suture it back together. What we'll usually do is kind of put a ostomy bag in there, right? Make sure that it heals up a little bit first, and then after, a little bit of time, reattach it together.
I watched the surgery for those.
Yeah, it's kind of crazy. All right,
Well,
we
they,
call it reanastomosis.
yeah,
But that can happen, and that's what we're worried about with diverticulum. Now, keep in mind, in the large intestine, right, you have this connection, right, the ileocecal junction. On that connection, you have the valve, which is the name of the sphincter, right? Then all the contents goes into the cecum, which is connected to the appendix. This is the ascending, right, colon, transverse, descending colon. Within the colon, you have pouches that we see called haustra. That's the right order. That's the nature of that inner circular layer, right? Now we see the flexures, which allows us to have a turn in the colon. The right colic flexure is also called the hepatic flexure because of the liver. The left colic flexure is called the splenic flexure because your spleen is up here, right? Keep in mind, main purpose now is to move that feces from one area all the way into the rectum. Once it gets to the rectum, you stretch the rectum, and now you feel the urge to go number 2. I know, right? So what do we see in the anal canal? The anal canal is a very end, simple columnar cells, and then towards the end where it comes out, all right, stratified squamish. Why stratified squamish? Because stratified squamish is protective. So let's take a look right here. Here's your anal canal. And then every now and then you might see what looks like little streaks right here. That's your hemorrhoids. Keep in mind, hemorrhoids are veins that are found on the outside right here. Veins right here that we can start seeing slightly. And then in some people who have a hard time going number 2, who are constipated, every now and then, right, the hemorrhoids can prolapse out, right? As it prolapses out, usually it's not a problem, it can go back in. The problem is if it gets stuck, right? That's where we have to use anti-inflammatories like preparation H, so then you can kind of decreases the inflammation, and then the hemorrhoid will come back into its original location, right? In the old days, they would, kind of rupture it, right? And if you rupture that vein, yeah, you might drain the hemorrhoid, but this is a very dirty area of the body, right? So it increases your chances of infection tremendously. Nowadays, we seldom, right, drain the hemorrhoid. We have an internal and external anal sphincter. The important thing to note is that the internal anal sphincter, smooth muscle, involuntary. The external anal sphincter is skeletal muscle, and it is voluntary. In other words, right, when we eat and fresh food goes into your stomach, the old food gets pushed from your small intestine. So you're having lunch, and now stuff from breakfast that was working its way through the small intestine. Well, we need to make room. So as you're eating lunch, we're sending the message to your brain. The brain is sending that message to your stomach and the rest of your GI tract, right? So you have that fresh food coming in. All the old food from breakfast needs to move from the small intestine, and we have bulk movement of that food into the large intestine, right? Once it gets in large intestine, it stays here, and all the older food gets pushed into the rectum. So then we can go number 2 and get all that old food out, right? Keep in mind, there's that we have to find a balance between keeping that food matter there so then we have increased absorption, but if it stays too long, it becomes a problem, right? You have feces that'll stay too long, and that's not good. Right, so in terms of large intestine, mucus provides protection. None of this is really highlighted because really, for the most part, the main purpose of the large intestine is to produce the feces, right? We will have water reabsorption and a little bit of vitamin K, which is absorbed here, but that's it. No more absorption of nutrients occurs once we go past the ileocecal junction. Right? Now, movements in the large intestines, we see mass movements common after a meal integrated by the enteric plexus. The mass movements are exactly what you're thinking. So mass movement is the overall sudden movement. That sudden movement integrated by enteric plexus, and it allows us, and it's, you know, some people are pretty embarrassed by it, but it's a normal thing, right? So if your boyfriend, girlfriend, husband, wife, both, right? If they have to go number 2 right after eating, it is because of this, right? So what happens is after you've eaten a new meal, the previous meal moves completely into the large intestine and then into the rectum. Crazy, right? These local reflexes in the large intestine instigated by presence of food in the stomach and the duodenum. Again, fresh food, fresh nutrients, tend to move everything out that we can. So the gastrocolic reflex initiated by the stomach activates mass movements when new food is introduced in the stomach. The duodenocolic reflex initiated by the duodenum also increases activity of mass movement, so then you can clear the duodenum, so then all the old food goes instead into the large intestine. Right, perfect. That's why you have to go poop it, right, after you've eaten. So if somebody's giving you a little bit of stick for, you know, going to the bathroom right after you've eaten, that's why. It's a reflex, nothing we can control. Right, so here it is, right? Present of food in the stomach, new food, then we stimulate mass movement, right? This is the gastrocolic. Then when the food goes in the duodenum, duodenocolic. Both allows us to have this mass movement. So then that old food goes from the distal ileum into the large intestine, into the rectum, which then causes a stretch of the rectum, which then sends that signal to your brain, and then your brain sends a signal to go number 2, right? So there is a time where you can hold off, right? So you guys have probably been on a freeway when you're younger, driven to a certain area, right, for vacation, and it's never convenient when you have to go number 2, right? So you just passed, you know, a gas station, or you just passed the last rest stop for like 20 miles, and then you feel the urge. Well, usually we can wait a little bit in terms of the defecation reflex, right? You do feel the urge though. It becomes worse and worse with time. As you have more and more stretch, you have greater and greater urge being ascending into your brain. Right. In terms of digestion and breakdown, this is a good slide because this one shows us what happens with carbohydrates. I wish I could make this smaller. Carbohydrates, lipids, and proteins. For carbohydrates, right, starts off being digestion with salivary amylase. So you take all, right, if it's going to be like starch, super long chains of glucose, the salivary amylase breaks it down to longer, all right, polysaccharides, numerous polysaccharides, but it's now shorter, right? Now, the salivary amylases do not work, though, on lactose, like in milk. The salivary amylases don't work on, right, other, all right, like on sucrose as well, right? So they only work on starches. They don't work on cellulose either. None of these work on cellulose, right? So these polysaccharides then go in the stomach where nothing happens. And then the duodenum, you have the pancreatic secretions containing amylase that chops everything off into disaccharides. Now, the disaccharides then gets broken down by brush border enzymes in the small intestine. So the brush border enzymes are your maltase, breaks down glucose and glucose. They are your lactase. Glucose and galactose are then separated. And then your sucrase, which then allows you to break down fructose and glucose, right? And separate them. You can digest each of the 3 monosaccharides. You can absorb each of the 3 monosaccharides. So we do see monosaccharides, right? You do see glucose, galactose, and fructose in the hepatic portal vein. But once those 3 monosaccharides get absorbed and goes in the liver converts everything into glucose. So the liver's main purpose is to take the fructose, convert it into glucose. Take the galactose, convert it into glucose. That's why you seldom hear of a blood glucose, blood galactose count. One time you do see it is when you might have a liver issue, and that's about it, right? Again, find your way through these in terms of fat, right? Lingual lipase. Now, gastric lipase is only seen in babies. So I'm just gonna, right, end this right here, and just keep that in mind that in terms of gastric lipase, right here, right, gastric lipase is seen in infants. And the reason why is simple, right? Infants, right? Infants eat lots of milk, right? Breast milk that is high in fat. So even the stomach in infants are different. Instead of producing pepsin, infants produce lipase because there's more fat in their diet, right? Then you have bile and pancreatic lipase that finishes it off. And the thing with fats is that you don't need a receptor. We just need to make sure that it's small enough that go through the plasma membrane. Fats like to react with other fats. So as long as it keeps separated, we can absorb it without a receptor. Proteins, no digestion in the mouth. It only starts its digestion in the stomach with pepsin, right? Which is produced by pepsinogen, right? Then in the small intestine, you cut it up. So then by the time you're done, right, you have single and diamino acids or dipeptides and tripeptides, right? Now, monosaccharides and amino acids and dipeptides and tripeptides, they go directly into your bloodstream. Fats goes into your lacteals first. So there's a difference in how they get absorbed, right? So here's the sugars, here's glucose coming in, right? In order to bring glucose in, you need a little bit of sodium coming in to the cell as well. We call this a sodium glucose co-transporter. What really happens is this. We have a secondary active transport pump. So this is found in our intestinal cells, the simple columnar cells with lots of microvilli. We look at one cell, here's the side facing the food. Here's the side facing the capillary and the lacteals. We've broken everything down. Now we have monosaccharide of glucose. This can easily be galactose or fructose as well. We have receptors for both of those other 2 monosaccharides. And both of them require sodium to go in. By the way, this is why Gatorade works the way it does, right? So what happens, right? Why is sodium so important in bringing it in? It's because of the secondary active transport pump. On one side of the cell, you see a sodium-potassium pump. You pump out sodium, pump in potassium. So what happens if you keep pumping out sodium? Inside the cell will be very low sodium, right? Very, very low sodium. So low that when we eat, the food has a lot of sodium in it. That sodium now creates a concentration difference. In the lumen of your GI tract, let's say you've eaten a saltine cracker, right? Saltine has a lot of salt, sodium, and has starch in it. So what happens? The sodium concentration is going to be very high in our chyme, right? Because it's so high, sodium wants to go from the outside of the cell to the area of lower concentration inside the cell. So as sodium goes inside the cell, it brings glucose along with it. We call this the sodium glucose co-transporter. And again, what do we see in Gatorade? Right? Electrolytes or salt and lots of glucose, right? What happens then? Well, the salt allows us to bring the glucose. As salt and glucose goes through, they're both, right, solids. So water wants to follow, right? So now you have salt, glucose, and water, right, that you're absorbing and incorporating into the cardiovascular system. Perfect. Now, in terms of fats, all we have to make sure for fats is that they're small enough, right? Then we break it apart by the lipases. As the lipases from the pancreas breaks it apart, then bile coats the outside of the fat, so then it can't recombine. So now it's small enough. The fat globules are small enough they can go right through the cell. As they go right through the cell, this fat, remember, fats like to react with other fats. So it goes right through the plasma membrane made out of phospholipids, and then we have the fats inside the cell. Now, the last thing we want to do is just put fat into any vascular system, right? We don't want to put it into the lacteal, but right? The reason why is simple. If I keep putting fat into the lacteals, the fat, even though they're small now, well, they're going to combine in the lacteal. As they combine, they form a fat emboli, right? Fat and water doesn't mix. So when you just have fat and we don't have any protein in it, and it's just fat being introduced, well, those small particles of fat becomes big particles of fat because they like to combine with each other. We need to prevent it. We prevent it by putting a protein coat in there. Just a little bit of protein, and the main purpose of that protein is to make it more soluble in our plasma, right? The thicker the protein coating, the more soluble it is, the better it is. The more soluble it is, the less likely it's going to precipitate out and form a blood clot, a fat emboli. So what we see here when we absorb it, we take a very small protein coat. As we put a small layer of protein coating on it, right? Very, very small, like 1% protein. That's it. Enough though to make sure that it doesn't precipitate. As we put that protein coat in it, we call it a chylomicron. So chylomicrons are then introduced into the lacteal. It's got a little bit of protein, 1%, and a lot of fat, right? But by putting that protein in there, it makes it slightly soluble. So then it doesn't recombine with other chylomicrons. And then as it goes into the thoracic duct, it goes into the left subclavian vein, and then eventually into the liver. The liver will take the chylomicron and then transport it as something else that we'll talk about next chapter, right? So these chylomicrons, 1% protein. They enter the bloodstream at the junction where the thoracic duct meets the left subclavian vein, right? In the bloodstream, it then goes to the liver, right? The liver then takes that chylomicron and then turns it into one of these, right? So all lipids are carried in blood are done so in combination with proteins. Adding more and more proteins makes them more soluble in plasma, making it less likely to have a fat emboli, or, God forbid, even a thrombus, right? Cholesterol, 15% are ingested, 85% are manufactured in the liver and intestinal mucosa. In other words, we can have people with very high cholesterol, even though they might not eat a lot of it through diet, right? Lipids are lower density in water, proteins are higher density. When we add it together, right, when we have fats and we add fats and then we add proteins to it, right? By itself, lipid, very low density, doesn't want to mix. By adding protein, we make it more dense and more soluble. Perfect. That's why HDL, high density lipoprotein, it's got a lot of protein, right? Making that less likely to form a thrombus or clot. This is our good cholesterol. So if you heard of good cholesterol, that's it, HDL, right? Good cholesterol, high protein. The other two here are the bad cholesterol. Why are they bad? Because it has less and less protein. making it more likely to diffuse out, making it more likely to form a thrombus or fat emboli. LDL, low density lipoprotein, 25% protein, right? This is our way of transporting cholesterol to the cells, which is perfect, right? So then, we have excessive amount of fat in our diet. What happens, right? The fat gets transported as chylomicron, goes into the liver, and the liver takes a chylomicron and then turns it into one of these, right? Very low density or low density, right? So LDLs, low density, are found in the liver and in your bloodstream after the liver has received the chylomicron. It transports cholesterol to the cells. The cells use the cholesterol as part of the plasma membrane. The cell uses the cholesterol in certain areas, like in the ovaries and the testes. The cholesterol is used to produce testosterone and estrogen, right? So keep that in mind that we do have uses of the cholesterol and the fats. It's not just always a bad thing. But it is bad if all we have are VLDLs, right? VLDLs has such high lipid concentration that it can form clots. So we know that if you have high amounts of this, there's a chance that you're more likely to have an MI, right? Or a stroke from fats that deposit into the unbroken blood vessel. Almost done. All right, amino acid transport is very similar to what we see in glucose, right? You're broken it down here. Now we have a single amino acid. Amino acids use secondary active transport, right? Just like in glucose. We pump out sodium by the ATP pump. The ATP pump doesn't directly cause this effect. What the ATP pump does, though, is creates a low sodium environment. That low sodium environment means that sodium from the outside, dietary sodium, that's in the lumen of our GI tract, that dietary sodium, very high in sodium, wants to go into area where there's low sodium in the cell. The cell has low sodium because of this pump. So that pump gives us a low sodium environment. Now sodium wants to come in. As it comes in, it brings amino acid along with it, right? This is a sodium amino acid co-transporter. As amino acid goes in the cell, it just goes right through into the capillaries, right? Just like glucose, just like galactose, just like fructose. So they go right into the capillary, which then goes to the hepatic portal vein and to the liver. Liver gets first dibs on all nutrients, right? That's it. In terms of effects of aging, you guys know what I'm going to say, right? It's going to slow down, it's going to shut down, and it's going to suck. So don't get older. In terms of, waters and ions, take a look at the numbers here. This is what I wouldn't ask you direct numbers, but it's crazy how much liquids that we have in our GI tract. Through ingestion, at least 2 liters a day. Then 1 liter of saliva, so far that's 3 liters. 2 liters of gastric secretion, 5 liters of secretions already, right? When you look at your bottle of Dasani, right? A bottle of Dasani, like a 16 ounce, that's half a liter. So at this point, we have 4 bottles you've ingested, then an extra 2 bottles from salivary gland secretion, extra 4 bottles from the gastric secretion. That's 10 bottles of Dasani, right? Already. And then pancreatic secretion and bile secretion, that's another 2 liters, another 4 bottles, and another 4 bottles from small intestinal secretion. So if you take a look at it, that's a lot of fluid going through the GI tract every day. Only 1% of it is going to be released as feces, right? 99% absorption of that fluid. So that's amazing stuff. And the reason why is water, fluid is very, very, very important, right? It's more important than gold. So we can't afford to lose excess fluid in terms of feces. So we reabsorb 99% of that fluid, of that water that goes either through ingestion or through secretions. Perfect. All right. So tomorrow I will be on here at like 12 o'clock, maybe a little bit earlier, 11:30 if I can. All right. So then we'll have a little bit more time. If you guys have any questions, feel free to ask. I probably won't go into any new material tomorrow. So you two are free to go, right? I will just have like a review section tomorrow. Review, if you guys have any questions on the actual models that you saw online. I do have some of the liver to stomach models I can show you by, opening and doing the, showing the camera. Okay?
Is that what's going to be on this one that's coming up tomorrow, or this is for the next one?
What's that?
Like the models of the liver and things, that's not going to be for this one that you're
I believe it is.
Oh, it
It's
is?
for this one.
Okay.
All right. So because the next one's going to be smaller. All right. The final exam will be smaller.
Okay.
And to make sure that it's smaller, this one's a little bit bigger because it's, we have more time for this one too.
Yeah. What is so we're going to have obviously this one opening up tomorrow, the lab practical.
Mm-hmm.
You mentioned like a quiz. There's going to be a quiz in between the final exam.
Yeah, there's a quiz on, and there's a reason why. There'll be a quiz on chapter 25 in terms of the actual, right, steps in Krebs cycle, glycolysis, and specific steps, right, will be on the quiz in chapter 25.
Okay.
I don't want you to memorize all the steps for the test. It's just a lot of steps, right? So for all the reactions, it'll be in this form of a quiz instead.
Okay.
So then you'll have your notes there, and it's not quite as intense for the test.
All right. And then you've recorded some of these. Like, are you posting them?
Yeah, I posted the other one. I will post these coming up.
Where do you post those?
I'll link to it on YouTube.
Okay, perfect. All right.
All right. Questions, Stephen?
No, I'm okay right now. I'm just foot's bothering me now a little bit. Sitting too long. My foot's bothering me sitting like this.
Would you go? All right.
I gotta go wrap it. I gotta go kick it up.
What part of your foot?
The top.
Did something fall on it?
No, it was a really awkward fall that I had in there.